Hi everyone, I am Dezso Modos from Norwich. I am working in the Erlem Institute and the Quadram Institute in the Kocsmáros lab. And I would like to talk about our viral link workflow. So we are using this workflow to fight against the coronavirus and figuring out that how the virus will interact with the human cells. First, let's know the enemy. We know that SARS-CoV-2 caused already more than 400,000 deaths worldwide and 43,000 in the UK. The virus itself enters both receptor-mediated through the ACA and this co-receptor PMPRSS2 to the cell, and both receptor-mediated transport. Then inside the cell, it releases its RNA, and then it's translated to proteins. These proteins and its fragments will interact with various human proteins and there propagate the signal to the various transcription factors and induce a transcriptional response. But what are these pathways which are involved in the host? What are how the virus hijacks the human cells? To answer these questions, we have developed a viral link workflow. The viral link workflow connects the interactions, the viral protein-human interactions with the gene expression results. To do that, we took the virus-human interactions from the Gordon et al. Data sets through the INTACT database. Then we used the Blanco Elo et al. Human bronchoepithelial cells gene expression data. From them, we found the differentially expressed genes. These differentially expressed genes we mapped back to transcription factors using the Dorothea database. Then, we filtered as well the signaling network for those genes which are expressed in these bronchoepithelial cells. Then we get a condition-specific PPI. So now we have the viral proteins, the interactor partners and the signaling network, then the transcription factors and the target genes. But we need to select that which is the shortest path between them. To do that, we use the algorithm called tie-dye, which is a network diffusion algorithm. It basically diffuses from both directions, both the transcription factors and the interactor partners of the virus, and tries to find the best shortest passes and also which are heavy in the best signs in the network. After that, we have done a functional clustering and centrality analysis in this network and also visualized it and seen that what are the pathways in the network. Let's see that how it looks like. Here we have the viral proteins, their interactor partners from the Gordon et al., the signaling network and the transcription factors and its target genes. We can see that it's much bigger blue blobs, so more signaling proteins are involved in the network compared to what we have originally. That's what we basically expect. But what are these transcription factors and differentially expressed genes? There are many which are both actually. One of the examples are the FOXO1 and the NF-kappa B, here one protein. Then we also want to look at which are the central parts of this network. To do that, we use the between a centrality measure. It measures the shortest passes which is going through for a certain node. We found various kinases, such as the CMP-dependent protein kinase, the ACT1, MAPK, MAPK3, which are very central kinases involved both in cell proliferation and apoptosis. Besides that, we found various transcription factors, such as the NF-kappa B, which is the central transcription factor involved in inflammation, and everyone's favorite, the TP53. Then we also looked at which pathways they are involved, which gene ontology functions. We looked at both the differentially expressed genes and the network itself as well. In the differentially expressed genes, we can find the interleukin signaling and various other immune-related signaling, such as the immune system. This is what we expect basically as a signaling link part which will be induced by a virus. But what is upstream? Not so surprisingly, we found the same enriched functions, again, like just cytokine signaling or signaling by interleukins, but also various tyrosine kinases, which is also expected, seeing the more central nodes. Then the toll-like receptor signaling, which is one of the viral protein recognition system in the human cells, and also the platelet activation and the aggregation, which is part of the coronavirus effect, because coronavirus patients have blood coagulation diseases, and their blood coagulates much quicker. So in conclusion, we have built up the ViralLink workflow. Its use is only a transcriptomic dataset, and you can get all of that which I have shown you within one click in a Python interface through our GitHub repo. It does not need anything else, and it generates much more additional figures as well. In using the Blanco-Melo et al. Dataset, we have shown that there are various kinases, such as the ACT, the MAPK kinase, and the CMP-dependent protein kinase, which are involved in the coronavirus, and also various transcription factors, such as the NF-kappa B or the FOXO. And we've seen that the signaling which is involved is the toll-like receptor signaling, the various immune and cytokine signaling, and also the blood coagulation. So thank you very much for your kind attention, and I am looking forward to see you on GitHub, and I hope that you will check out our ViralLink workflow. Thank you.